What if the cure for inflammation, premature aging, and cellular decay wasn't in a supplement bottle or behind a prescription pad, but had already been written down thousands of years ago in the most widely read book in human history? And what if peer-reviewed science, published in clinical journals, conducted at Harvard, EPFL, and the NIH, kept arriving at the exact same conclusions that ancient text already described? Not metaphorically, not spiritually, biologically. Most people are spending hundreds a month on collagen powders, NAD+ boosters, and longevity supplements, compounds isolated in a lab, stripped of everything that made them work and sold back in a capsule. Meanwhile, entire civilizations, people who built empires, crossed deserts, survived famines, and displayed a physical vitality modern medicine is only now measuring, were eating these exact foods. Whole, prepared the way tradition taught them, combined the way centuries of use refined them. The supplement industry found one compound and called it a breakthrough. These ancient texts described the whole food. And the whole food, it turns out, is exactly what the science keeps coming back to. And I want to be precise about what I mean when I say science keeps arriving at the same conclusions. Not folklore. Not wellness blogs. Not ancestral health influencers. Randomized, double-blind, placebo-controlled clinical trials. Skeletal muscle biopsies. Peer-reviewed molecular research indexed by the NIH. Studies conducted at the Swiss Federal Institute of Technology EPFL and the George Spirehouse Institute in Frankfurt, specifically measuring what these foods do at the cellular level. What researchers found repeatedly across independent studies and independent populations is that the bioactive compounds in these foods don't just provide nutrition. They function as signaling molecules. They interface directly with the genetic switches that control how fast your cells age. Switches called SIRT1, AMPK, and mTOR. The same pathways every serious longevity researcher in the world is focused on right now. This is not promising science. This is not emerging science. This is documented, replicated, and in several cases the current scientific consensus on how cellular aging works, and what slows it down. And the foods at the center of it were described thousands of years before any of these researchers were born. Today I'm walking you through 10 foods that appear repeatedly across the biblical text. Foods that sustained prophets, fed armies, and sat at the center of the most significant meals in recorded ancient history. And what modern science has confirmed about each one is specific, measurable, and in several cases, genuinely remarkable. Here's what we're covering. Cellular senescence, where damaged cells stop dividing but refuse to die, quietly accelerating aging in every tissue around them. Mitochondrial decay, the breakdown of the energy-producing structures inside your cells that drives fatigue, cognitive decline, and accelerated physical aging. Number 7 is a seed. It sits on the bottom shelf of most health food stores, largely ignored. And what modern microbiome and hormonal research has confirmed about it is extraordinary. It contains a compound your gut bacteria convert into a molecule so structurally similar to estrogen that it can modulate hormonal inflammation across the entire body. Clinical meta-analyses show it significantly reduces 2 of the most important biomarkers of chronic systemic inflammation in circulation. Most people walk past it every single week. Number 10, stay for this one specifically, was prescribed in the biblical text for physical exhaustion and recovery. And what it contains maps almost exactly to what sports scientists and longevity researchers now identify as the most critical nutrient for protecting the chromosomes in your cells from damage that accumulates silently over decades. There is a 5-year observational study of cardiac patients, and a series of controlled trials, that tell a story most people in the health space have never connected back to where it first appeared in the historical record. We're starting at number 1 right now. If you want biblical nutrition and ancestral health wisdom, backed by peer-reviewed molecular research every single week, hit subscribe and ring the bell right now. These videos don't show up in your feed unless you tell the algorithm you want them. So if this content matters to you, make sure it finds you every time. Let's get into it. Here are the 10 biblical foods modern science has confirmed slow aging at the cellular level, starting with number 1. 1. Pomegranate. the ancient fruit that teaches your cells how to clean house. When Moses sent spies into Canaan, they returned carrying three things as proof of what the land could produce: grapes, figs, and pomegranates. Not grain. Not meat. The pomegranate appears across the biblical text not as background detail but as something valuable enough to be woven into the hem of the high priest's robe, carved into the pillars of Solomon's temple, and named one of the 7 species of the Promised Land. Modern molecular biology is now explaining why. Pomegranate contains some of the highest concentrations of elagitannins, complex polyphenol, of any food ever studied. When you eat it, your gut bacteria metabolize those elagitannins into a compound called urolithin A. And urolithin A does something no other food-derived compound has been clinically demonstrated to do at this level of specificity. It activates mitophagy, the process by which your body identifies damaged mitochondria and clears them out, forcing the regeneration of healthier ones in their place. Clinical trials confirmed this in human skeletal muscle biopsies. In a randomized, double-blind, placebo-controlled trial, adults taking 500 milligrams of urolithin a daily showed significant upregulation of the mitophagy pathway in skeletal muscle. At 1,000 milligrams daily, a 20% increase in muscle fatigue resistance across anatomically distinct muscle groups. No change in exercise habits. Separate trials showed urolithin A reverses key markers of immune aging by expanding the naive CD8 positive T-cell populations. Your immune system depends on to fight threats it has never seen before. One critical detail: only 30 to 40% of people have the gut microbiome required to convert pomegranate's elagitannins into urolithin A. The majority eating pomegranate are not getting its most powerful effect. We'll come back to why that matters when we talk about how these foods work together. Traditional preparation was direct and whole. Seeds eaten fresh, juice pressed and consumed immediately. Nothing extracted. Nothing isolated. The entire food matrix intact. Modern research is confirming the matrix matters as much as the molecule. 2. Olives and extra virgin olive oil. The oil that tells your muscles not to age. The olive tree is one of the most recurring presences in the entire biblical narrative. The branch the dove returns with after the flood. The source of oil used to anoint kings and priests. The Mount of Olives framing the most significant events of the New Testament. Olive cultivation was the economic and nutritional backbone of the ancient Near East. So central that its destruction in warfare was considered one of the most devastating acts an army could commit against a civilian population. What those ancient populations were consuming daily, modern biochemistry has now identified with precision. Extra virgin olive oil contains a dense phenolic fraction dominated by a compound called oleuropein-aglycone, OLE. In human skeletal muscle cells exposed to oxidative stress, OLE reduced intracellular reactive oxygen species by approximately 43% and cut the accumulation of stress-induced senescence cells by 12%. It does this by activating a signaling cascade that upregulates the body's own antioxidant enzymes and increases the expression of stress-protective proteins called sestrins. Simultaneously, OLE activates autophagy through AMPK-mediated suppression of mTOR, triggering the cellular cleaning process that clears damaged proteins and dysfunctional organelles before they accumulate into the debris that drives accelerated aging. In neurodegeneration models, EVO polyphenol cross the blood-brain barrier and promote clearance of amyloid beta and tau proteins, the two hallmark accumulations of Alzheimer's disease. Hydroxytyrosol, another key phenol, increases neural stem cell survival in the brain's dentate gyrus and reduces the microglial activation that drives neuroinflammation. Clinical studies consistently link high EVO consumption to lower rates of cardiovascular disease, cognitive decline, and all-cause mortality. The benefit is real, measurable, and specific to genuine cold-pressed extra virgin oil. Not the refined, blended, partially adulterated product that makes up most of what sits on grocery store shelves under that label. Traditional use was simple. Cold-pressed. Consumed fresh. Used liberally. Never heated to the point of phenolic degradation. The ancient populations using it weren't thinking about bioavailability. They were thinking about flavor and abundance. The outcome, as the research now shows, was the same. 3. Figs. The fruit that outperformed metformin in diabetic models. When Hezekiah, king of Judah, developed a life-threatening illness, the remedy prescribed was not prayer alone. The prophet Isaiah instructed that a poultice of figs be applied directly to the wound. Most readers focus on the miraculous element of the story. They miss what the food itself represents. Figs were the first-line therapeutic food of the ancient Near East. The significance for cellular aging is direct. Chronic glucose surges are one of the primary drivers of advanced glycation end product formation. The cross-linking of proteins and DNA that stiffens tissue, accelerates vascular aging, and drives mitochondrial dysfunction. By blunting those surges, fig extract isn't just managing blood sugar. It's interrupting one of the fundamental mechanisms through which the body ages faster than it should. Traditional preparation preserved the whole fruit, dried slowly, eaten as is, combined with grain and oil. Nothing extracted. No compound isolated beyond its natural concentration. The whole food, delivering the whole effect. 4. Dates. The fruit that physically blocked DNA damage in laboratory conditions. Dates are perhaps the most practically embedded food in the entire biblical and ancient Near Eastern dietary record. Provisions for desert travel. Offerings. The sweetener of last resort in communities with no access to refined sugar. A food specifically noted for sustaining physical endurance under extreme conditions. Ancient armies measured the wealth of a conquered territory partly by the number of date palms standing in it. What dates contain at a molecular level is a dense matrix of phenolic compounds: gallic acid, catechin, quercetin, ferulic acid, alongside carotenoids, phytosterols, dietary fiber, and the essential trace element selenium. And what those compounds do to DNA has been demonstrated in direct laboratory conditions with a specificity that is difficult to overstate. In gel electrophoresis assays, researchers exposed pure DNA to a Fenton reagent, hydrogen peroxide and iron sulfate, that generates the hydroxyl radicals responsible for the most destructive oxidative DNA damage that occurs inside aging cells. Without intervention, complete DNA strand destruction. When date extract was introduced at concentrations of 10, 50, and 100 milligrams per milliliter, no destruction. The DNA maintained a clear, intact band. Complete protection across every tested concentration. At 1.5 milligrams per milliliter, date extract achieved 100% superoxide radical scavenging, outperforming ascorbic acid, which showed zero scavenging capacity at the same concentration. Beyond DNA protection, date-derived phytochemicals suppress NF-kappa B, the transcription factor driving the chronic inflammatory gene expression program at the center of what researchers now call inflammaging. In mouse models of accelerated aging, inhibiting this same pathway delayed cellular senescence, reduced oxidative DNA damage, and improved tissue homeostasis across multiple organ systems. In the ancient world, dates were eaten as they came off the tree whole, fresh, or sun-dried. Combined with water, olive oil, or grain. Nothing removed. The full phenolic matrix, the fiber, the selenium, all present. All working together. The way the food was designed to deliver them. 5. Garlic. The compound that keeps your mitochondria from falling apart. When Roman soldiers marched across 3 continents, they carried garlic in their rations, not for flavor, for function. The same practice appears across ancient Near Eastern armies, Egyptian labor records, and early Ayurvedic medicine. Garlic was the performance food of the ancient world, used instinctively by civilizations that had no mechanism to explain why it worked. Only thousands of years of evidence that it did. Modern molecular biology has now identified the specific mechanism. Aged garlic extract contains high levels of a water-soluble organosulfur compound called S-allylcysteine SAC. And SAC does something that places it in a category almost entirely its own in the nutritional literature. It directly regulates mitochondrial dynamics. During normal aging, the balance of mitochondrial activity shifts toward excessive fission, the splitting of mitochondria into smaller, fragmented, dysfunctional units. This is not a trivial cellular event. It is one of the primary upstream drivers of the energy decline, cognitive deterioration, and physical degeneration associated with aging. Maintaining a healthy mitochondrial network requires continuous fusion, the rejoining of mitochondrial membranes that allows damage to be diluted across the network. SAC restores that balance. In naturally aged mice, SAC supplementation significantly upregulated OPA1, the primary protein responsible for inner mitochondrial membrane fusion. By promoting fusion, SAC preserved the interconnected morphology of the mitochondrial network and drove a downstream increase in SIRT1 and PGC1-alpha, the master regulators of mitochondrial biogenesis. Cellular senescence markers were significantly reduced. SAC additionally activates NRF2, the master antioxidant transcription factor that induces the body's own protective enzymes, glutathione S-transferase, catalase, superoxide dismutase. In neurological models, SAC protected dopaminergic and cortical neurons from oxidative damage by suppressing endoplasmic reticulum stress pathways. One preparation detail matters. Crushing or chopping garlic activates the enzymatic conversion of alliin to allicin. Further aging or slow fermentation produces the stable, water-soluble SAC the research consistently identifies as the most bioavailable and therapeutically potent form. The ancient practice of aged or fermented garlic was, biochemically, exactly the right preparation method. It just took modern science 2,000 years to explain why. 6. Lentils. The forgotten legume that activated the same cellular switch as caloric restriction. When Esau returned from the field exhausted, depleted, physically spent, and traded his birthright for a single bowl of lentil stew, most readers focus on the impulsiveness of the exchange. They miss what the food itself represents. Lentils were not a consolation meal. They were the caloric and nutritional backbone of the ancient Near East, the staple of laborers, soldiers, and travelers precisely because they were among the most sustaining, most restorative foods available in that landscape. The exchange Esau made was culturally equivalent to trading something permanent for something that, in that moment, his body recognized as exactly what it needed. Modern nutritional science is now explaining why the body makes that recognition. Lentils contain a dense matrix of bioactive polyphenol that target the AMPK signaling pathway, the same cellular energy sensor that caloric restriction activates. The same pathway every serious longevity researcher identifies as one of the most important metabolic switches governing how fast cells age. When activated, AMPK initiates a cascade that stimulates fatty acid oxidation, enhances glucose uptake, suppresses energy-consuming anabolic processes, and promotes autophagy and mitochondrial quality control. Population studies consistently link high legume consumption to lower rates of cardiovascular disease, colorectal cancer, and cognitive decline. And across every long-lived population ever formally studied, Sardinia, Okinawa, the Nicoya Peninsula, legumes are the single most consistent dietary feature. Not meat, not dairy, not supplements. Legumes. Lentils also carry one of the most favorable potassium to sodium ratios of any whole food, approximately 30:1, making them a structurally significant contributor to vascular health across a lifetime of consumption. One preparation detail the ancient world got exactly right. Overnight soaking before slow cooking. That step reduces phytic acid and enzyme inhibitors that otherwise block absorption of the iron, zinc, and magnesium lentils contain. Modern nutritional science calls this improving bioavailability. Ancient cooks called it how you prepare lentils. The outcome was identical. 7. Flaxseed. The seed most people walk past, and what it does inside your body will surprise you. Here we are at number 7. The one I mentioned at the top. Flaxseed. A seed so unremarkable in appearance, so consistently relegated to the bottom shelf of health food stores, that most people buying it have no clear idea what they are actually purchasing. They know it is supposed to be good for them. They do not know why. What modern microbiome and endocrinological research has now confirmed is genuinely one of the more remarkable findings in nutritional science of the last 2 decades. Flaxseed is the richest known dietary source of a plant lignin precursor called seco-isolariciresinol diglucoside, SDG. It provides up to 800 times more lignins than any other oilseed, grain, or vegetable. When you consume flaxseed, your colonic microflora metabolize SDG into 2 bioactive compounds, enterodiol and enterolactone. Phytoestrogenic molecules structurally similar enough to estrogen that they can bind to estrogen receptors and modulate hormonal signaling throughout the body, with particular relevance to inflammation regulation, metabolic function, and cellular aging. Clinical meta-analyses of randomized controlled trials confirmed that flaxseed intake significantly reduces 2 of the most critical circulating biomarkers of chronic systemic inflammation: high-sensitivity C-reactive protein and tumor necrosis factor alpha. Among the most validated predictors of accelerated biological aging in the published literature. At the molecular level, SDG extract downregulates the hedgehog signaling pathway, a developmental pathway that, when chronically overactivated in adulthood, drives adipose tissue inflammation, metabolic dysfunction, and the lipid accumulation increasingly understood as a driver of cellular senescence. SDG suppression of this pathway regulates lipid homeostasis, decreases hepatic lipid accumulation, and improves peripheral insulin sensitivity. It also modulates autophagy-related genes, promoting clearance of lipid-laden cells and protecting against obesity-related cellular stress. The biblical record does not name flaxseed with the prominence of olive oil or pomegranate. But linen, made from the flax plant, is one of the most consistently mentioned materials in the text. From the garments of Egyptian priests to the fabric of the tabernacle. Where linen was grown, flaxseed was eaten. A food hiding in plain sight in the agricultural record of the ancient world. One preparation detail that is not optional. Whole flax seeds pass through the digestive tract largely intact. Ground flaxseed is what delivers the lignins to the microbiome that converts them. That detail, ground, not whole, is the difference between eating flaxseed and actually receiving what the research measured. Wheat germ, the part of the grain they removed 100 years ago, and what disappeared with it. This is the one that surprises people. When industrial milling became dominant in the late 19th century, the wheat germ was removed from the flour. The stated reason was shelf life. Wheat germ contains oils that go rancid, shortening commercial viability. The unstated consequence was the removal of one of the most concentrated sources of a compound that modern longevity researchers are now calling a physiological autophagy inducer equivalent in potency to rapamycin, the FDA-approved immunosuppressant currently one of the most studied life extension compounds in geroscience. That compound is spermidine, and wheat germ is its richest dietary source. Spermidine is a naturally occurring polyamine present in every living organism. Tissue and blood concentrations decline in a measurable, age-dependent manner in both rodents and humans. This decline directly correlates with a reduction in systemic autophagic capacity, the body's ability to clear damaged proteins, dysfunctional organelles, and cellular debris that accumulates with age. There is one documented exception. Healthy nonagenarians and centenarians maintain blood spermidine levels comparable to individuals decades younger. The correlation between high spermidine status and extreme longevity is one of the most consistent findings in the human aging literature. Mechanistically, spermidine induces autophagy by inhibiting a family of acetyltransferases, most critically EP300. EP300 acts as a brake on the autophagy machinery by acetylating lysine residues on core autophagy proteins including ATG5, ATG7, and LC3, rendering them inactive. Spermidine removes that brake, maintaining these proteins in their active state, stimulating the formation of autophagosomes, the cellular structures that engulf and degrade damaged material. In a phase 1 clinical trial, 6 milligrams of wheat germ-derived spermidine daily in adults over 65 significantly enhanced CD8-positive memory T-cell responses during vaccination. Spermidine restored autophagic flux in elderly T-cells, improving vaccine-induced antibody responses and reversing measurable markers of immune aging. In separate in vitro models, spermidine inhibited SARS-CoV-2 viral replication by up to 85% by counteracting the virus's mechanism of suppressing autophagy, without inducing any cytotoxicity in host cells. Wheat germ appeared in the ancient diet because ancient milling was imprecise. Stone-ground whole grain retained the germ. The most nutritionally active part of the grain was consumed by default. It took industrial precision to remove it, and another century to understand what had been lost. 9. Olives revisited. The brain connection most anti-aging conversations miss. We covered olive oil at number 2. But what it does to the aging brain is specific enough and significant enough that it earns its own entry on this list. The research on hydroxytyrosol keeps returning to one target: neuroinflammation. The chronic, low-grade inflammatory activation of the brain's immune cells now understood to be one of the primary upstream drivers of cognitive decline, not just in Alzheimer's disease, but in the normal aging trajectory of the human brain. Hydroxytyrosol crosses the blood-brain barrier. Once inside the central nervous system, it increases the expression of LC3 and Beclin-1, the autophagic markers responsible for clearing amyloid beta and hyperphosphorylated tau. It upregulates SIRT1 in neural tissue. It directly inhibits both the NF-kappa B inflammatory pathway and the NLRP3 inflammasome, the two primary intracellular signaling complexes driving the sustained neuroinflammatory response associated with accelerated brain aging. In 5xFAD Alzheimer's mouse models, oleuropein supplementation enhanced blood-brain barrier structural integrity, suppressed inflammatory cytokine secretion, and improved cognitive function on behavioral assessments. In the BTG1 null mouse model, designed specifically to study neural progenitor depletion, hydroxytyrosol increased neural stem cell survival in the dentate gyrus, reduced lipofuscin accumulation, and decreased the microglial immunoreactivity that marks chronic neuroinflammatory activation. The ancient world had no concept of neuroinflammation, but it had a concept of mental clarity, of wisdom preserved into old age, of the mind remaining sharp when the body slowed. And the food most associated with anointing, with consecration, with marking someone chosen for a role requiring clarity of thought was olive oil. That connection was intuitive then. It is now molecular. 10. Fish. The food prescribed for physical recovery in Scripture, and what it does to the clock inside your chromosomes. Here we are. Number 10. Let me be clear before we go into this. This is not a religious argument. This is not a claim that Scripture is a nutritional manual. It is an observation about why the food that appears in the biblical text in specific contexts of physical restoration and recovery happens to be the food that modern longevity researchers have now linked, in randomized controlled trials and long-term observational studies, to the preservation of the structures at the tips of your chromosomes that determine how fast your cells age. Those structures are your telomeres. And the food is fish. Fish, specifically the long-chain omega-3 fatty acids EPA and DHA, protects telomeres through two distinct, independently validated mechanisms. The first is anti-inflammatory and antioxidant. Systemic inflammation and oxidative stress accelerate the loss of telomeric DNA during cell division. In a randomized, double-blind, placebo-controlled trial, middle-aged overweight sedentary adults receiving either 1.25 or 2.5 grams of omega-3 daily for 4 months showed significant dose-dependent decreases in inflammation and oxidative stress markers. A lower omega-6 to omega-3 ratio in the blood was associated with longer leukocyte telomeres, suggesting that shifting this ratio actively slows the rate of telomeric attrition. A separate 5-year observational study of coronary artery disease patients confirmed that higher baseline EPA and DHA levels were associated with measurably slower telomere shortening over time. The second mechanism is telomerase preservation under stress. Telomerase is the enzyme responsible for synthesizing new telomeric repeats. In the most literal biological sense, the body's chromosome repair system. In clinical stress tests, individuals not supplementing with omega-3 showed a 24% decline in salivary telomerase activity approximately 2 hours after acute social stress. Participants receiving either 1.25 or 2.5 grams of omega-3 daily did not experience this decline. The omega-3 had buffered the stress-induced suppression of the repair system entirely. High-dose omega-3 at 2.5 grams additionally lowered post-stress cortisol by 19%. What this means in plain language: omega-3 fatty acids from fish don't just reduce inflammation. They preserve the body's capacity to repair its own chromosomes under the conditions stress, oxidative load, physical exertion that most rapidly accelerate chromosomal aging. They keep the repair system running when the body most needs it, and is most likely to lose it. In the biblical account of the post-resurrection meal on the shores of the Sea of Galilee, the food prepared was fish. In the feeding narratives, fish is paired with bread as the complete sustaining meal. In the prophetic tradition, abundant fish is a recurring image of restoration and provision. These were not arbitrary food choices embedded in the text. They were the reflection of a culture that had learned, across generations of lived experience, that fish was what the body needed to recover, to sustain, and to endure. Modern science just told us what was happening at the level of the chromosome while it did. If you haven't subscribed yet, hit that button now and ring the bell. Comment your thoughts. We post every week.